Hello, everyone. My name is Lindsay Shuck with Family Office Networks. Thank you for joining our FON webinar series today. We are featuring Lactocore. Dr. Askar Kuchimov will be speaking today. Askar is a biotech executive with 15 years experience in business development and venture capital. He has raised and managed a total of $45 million of funding to turn early stage projects into viable clinical stage drug development companies. At the end of the presentation today, we will have time for Q&A. Please be sure to send over your questions via chat at any point during the presentation so we can answer them at the end. Askar, over to you.
Thank you, Lindsay. Hi, everyone. My name is Askar Kuchimov, and I am the CEO of Lactocore, Inc. Thank you all for taking your time to join this webinar. We all know about COVID-19, and you're probably tired of hearing about it by now. But there's another silent pandemic that is sweeping the world that you don't hear a lot about in the media, yet it is bound to get even worse after the coronavirus, and it is bound to have long-lasting consequences. I'm talking about anxiety and depression. In today's webinar, you will learn about why novel drugs against depression and anxiety represent attractive investment opportunity, and how Lactocore is uniquely positioned to capitalize on this new wave of interest to psychiatry indications. Just to give you an idea about the scale of the problem, here are some stats about the patient populations in the US alone. Antidepressants are actually one of the most popular drugs in the United States. 13% of Americans take antidepressants, and majority of them take them for more than 2 years. And as you can see, there's tens of millions of people taking the drug, and the economic toll on the society is measured in hundreds of billions of dollars. But the most intriguing part, all these stats are pre-COVID-19. A recent poll by Kaiser Family Foundation has revealed a staggering 45% of people claiming increased anxiety and stress related to coronavirus, with 19% claiming major negative impact on the mental health. And if you noticed, major news outlets started publishing warnings about it. And the experts agree. Here we have a quote from Professor Ed Bullmore from University of Cambridge. His key message is that COVID is likely to have major impact on mental health now and into the future. And everybody needs to start thinking about that immediately. And he obviously means the scientific and the pharmaceutical community. But the biggest problem, however, is not that these conditions are so widespread. There are plenty of drugs out there on the market designed to help. The global market is $17 billion. The biggest problem is that the existing drugs have very serious issues. For example, let's look at the mainstream drugs for depression and anxiety. SSRIs, which stands for selective serotonin reuptake inhibitors, and benzodiazepines, denoted as BDs here on the slide. There is also a typical antipsychotics, or AAPs. First of all of them have unacceptable medical side effects, sexual problems, metabolic abnormalities, like people gaining weight when taking those drugs, and lots of cardiovascular risks. Benzodiazepines actually are even worse. Common side effects include sedation and abuse potential. You have heard about the opioid epidemic in the US. And if you look in the stats, you will notice that benzodiazepines constitute a third of the prescription drug abuse cases. So in addition, antidepressants such as SSRIs would normally take 4 to 6 weeks to kick in. So if the first dose doesn't work, the therapist would often switch a patient to another drug and wait for another month or so. And that can take months. And still, at the very end, in 30% of cases, these drugs end up not working at all. So the part of the reason is that these drugs are actually very old. They were developed 30 to 40 years ago Like benzos were first developed in the 60s. SSRIs antidepressants were developed in the 80s. And another mainstream drugs, atypical antipsychotics, were developed in the 90s. So literally, decades went on with really no novel drugs approved by the FDA. Only last year, after a long break, the agency has approved 2 drugs in 1 month. You can see them here. They were approved in March of 2019. Sketamine was developed by Johnson & Johnson, and it was approved for the treatment-resistant depression, which is a depression that could not be treated by the existing drugs. Sketamine works great, but the problem is it's a stereoisomer of the old anesthetic drug ketamine, which you may have heard about. It's a very well-known drug of abuse with hallucinogenic and dissociative properties. It's been first approved by the FDA back in the 70s as an anesthetic. Another drug, allopregnanolone, was developed by Sage Therapeutics and approved in a very narrow indication of postpartum depression. It is actually a metabolite of progesterone. So in reality, it's a steroid, it's a neurosteroidal drug that has certain effect on the brain among many other things that it does in the body. So sedation and even loss of consciousness are among the side effects. And because of these serious adverse effects, both drugs can be administered only at the certified medical facility, which obviously severely limits their use. So what does Lactocore offer as an alternative? First of all, we develop drugs that have new mechanisms of action that are distinct from the approaches that are used in the past. We rely on the latest understanding of the pathology of depression and anxiety to trigger new mechanisms for rebalancing brain networks that are distorted in depression. Excuse me. Secondly, we focus on peptides, which are essentially short compounds made of amino acids. Distinctive features of the peptides are their high selectivity, very low toxicity, and short lifespan. So which makes them very good soft modulators that are perfectly suited for triggering healing processes in CNS. Our discovery methodology is built on the 2 main elements that constitute our core expertise. The first is computer and aided drug discovery. Here we rely on a supercomputer power and apply our own advanced algorithms for 3D protein and peptide modeling, as well as scoring of the best hits. Once we zero in on the most promising target, we apply algorithms to screen millions of 3D peptide structures against key targets. Here you can see, for example, a 3D rendering of a crystal structure of one of our targets, mGluR5, which is a key receptor in the glutamatergic signaling system in the brain. The second core element of our approach is a workflow that we develop in-house to quickly and efficiently screen in silico leads by taking them directly in vivo into the animal behavioral testing, starting with zebrafish and followed by traditional testing in rodents. I'm not going to talk a lot about zebrafish here, but we'll just mention that this tiny fish is actually one of the widely used models in research and drug development, and for the good reason, as its DNA carries similarity to 70% of human genes. You may ask about the behavioral rodent models. Many people are actually asking us, how is it possible to model a depression in a rat? And so I decided to spend a few minutes to give you some basic principles on how this thing works. Is there actually a depressed rat? It turns out that it's impossible to completely mimic human depression in the rat for the very simple reason that they lack self-reflection and self-consciousness, and so would not be able to become sad, for example, or have suicidal thoughts. Plus, depression as a disease is a very heterogeneous condition with many behavioral and physiological parameters. So instead, scientists create specific phenotypes in the animals by subjecting them to a variety of stressful conditions. For example, one can cause a rat to experience anhedonia or inability to experience pleasure and evaluate it by how much less of sweetened water is consumed by the rat. Another example is of a phenotype is anxiety that has a variety of readouts, and one of them is suppression of appetite in the novel environment. But regardless of the actual test that's being studied, it is important that it must be influenced by the existing antidepressant treatment. And so the novel drug candidates can be evaluated and compared. So one of the interesting examples that I wanted to demonstrate today is a model of a post-traumatic stress disorder, or PTSD. I think everybody knows what PTSD is. So I won't go into explaining about this indication. It's pretty close to, it's distinct from depression and anxiety, but nevertheless, it's a closely related indication. So in case of testing our leading drug candidate, we have used a test called predator odor model of PTSD, where we used bobcat urine as a stressor for a rat. So the idea of the test is to provoke aversion or fear by exposing animal to a highly stressful stimulus, such as urine of a bobcat, and do so in one of those two chambers, such as the one that you see on the slide. In the rat's brain, then, this chamber becomes associated with the danger. And so even after you remove the actual danger from this chamber, the rat will prefer not to go there. And this is a very rough and basic idea for this test that's called condition-placed aversion. So here, I just wanted to quickly show how the real data looks like that was obtained in this predator odor test. So I want you to just focus on the graph part of it, and you will see the 3 bars, 1 negative kind of and 2 positive bars. The negative bar is the control animals. So these animals that were subjected to the stress, but they were never treated. And the 2 other groups, one was treated with a known drug, which is a small positive bar in the middle, and the other group was treated with our leading candidate, LCGA17, which is the positive bar, the blue bar on the very right. So what basically that means is that the rats who were treated with the drugs were able to come back to normal and sort of forget the bad experience in that chamber and spend more time in it compared to untreated animals who avoided the danger chamber even after the stressor was removed. And so you can see that LCGA17, our leading acid, has performed significantly better in this test compared to positive control. Which brings me back to LCGA17. I would like to mention some of the other animal models that we have done with it, and just to give you a better idea about the amount and the quality of behavioral animal model data that we have accumulated for this asset. This is a pretty complicated table, but let me just kind of quickly walk you through this. On the left, you see the animal models, and then you can see that in addition to the PTSD model, we have completed a comprehensive suite of depression and anxiety models. And the idea was to look at the performance of our drug from many different angles. One of the main ideas in this table is that we have seen the robust efficacy of our compound in both depression and anxiety-related tests, which points to the potential to use this drug in more than one indication. So at a minimum, we believe that we can see 3 indications in promising indications right now: anxiety, depression, and PTSD. And on the right side, it's important to note that LCGA performance was either comparable to or superior to the standard of care drugs that were mentioned earlier, such as SSRIs, benzodiazepines, ketamine, and allopregnanolone. So taken together, all these data strongly suggest that we may be looking at a next generation antidepressive and anxiolytic drug with truly blockbuster potential. And to complete the summary about LCGA17, we have listed here a number of other attributes of this compound that provide a very solid competitive advantage compared to other drugs in the field of depression and anxiety. So first of all, the fact that LCGA17 works through a novel mechanism of action by modulating key molecular targets such as GABA-A and VGCC, which stands for voltage-gated calcium channels. Both of these are implicated clinically in the neuropsychiatry disorders. And just for fun on the slide, you can see a three-dimensional rendering of the GABA-A receptor structure. For those of you who are curious, it's actually a chloride ion channel consisting of 5 subunits, 2 alpha, 2 beta, and 1 gamma subunit. You can see them a little bit different colors. And it happens to be a major element in the brain regulating inhibitory processes, which exist in a delicate balance with excitatory processes, resulting in a healthy brain functioning and connectivity. And it is the disbalance of these processes that leads to the loss of connectivity between various brain regions, which people believe is causing pathological conditions such as depression and anxiety. Very importantly to note here that we have not detected any side effects for LCGA17 that are typical for standard of care drugs. We have seen no sedation and no addiction potential, which are very, very limiting side effects for some of the existing therapies, as we have already mentioned. And so using LCGA17 as a successful example of our internal validation flow, we started applying the same principles and methodology to generate a pipeline of novel candidates against various targets. You can see them listed here on the left side. So in addition to GABA-A and VGCC, we have mGluR5, TrkB, NTSR1, and NLP1 as targets. So by influencing these targets with our peptides, we will be able to address other CNS-related indications, such as ADHD, which stands for Attention Deficit Hyperactivity Disorder, movement disorders, schizophrenia, pain, and more types of mood disorders as well. And of course, in our development plan, we have allocated part of the budget to move those earlier targets, candidates forward as well. So if you look at the top line, going back to the LCGA17, you can see that it's already entered so-called IND enabling studies in the CMC program, which is basically FDA mandated toxicology studies in manufacturing, which basically means that we are on the finish line towards our first clinical trial, and clinical trial being the trial in humans. So this will be a major milestone and a value inflection point for this program and for the whole company. So moving on to the development plan, here's a high-level view of what we intend to accomplish with LCGA17 using the funds that we're currently raising. Our goal is $10 million, of which we have $3 million already committed by the existing investor. The funds of this round will cover the formal preclinical trials, such as GLP tox and the manufacturing for the LCGA, as well as phase 1 clinical trials. Phase 1s are usually done in healthy volunteers, and we have it here in the middle as phase 1A trials. But in addition to this, we're also planning to test the drug in patients, which would be phase 1B trials you see on the right. And the idea here is to lower the risk of the program by trying to get an early efficacy signal, or either directly or through the use of biomarkers. I also wanted to point out that we are decided to test depression and anxiety, which are basically two different indications, in parallel here, since the animal data was looking great for both. And it was hard for us and our advisory team to prioritize just based on the animal data alone. And if you see at the bottom, you will see an expected timeline, which basically projects about 3 years for completion of these milestones. So what happens after we successfully complete the projected milestone for the Series A? Our intent is to prepare the asset for the acquisition by a larger pharmaceutical company. These pharma companies are usually very good at the clinical stages. And if you look at the slide, you see phase 1, 2, and 3. These are the clinical stages of the development. And the pharma companies are very good at commercialization of the drug, but they're not usually so good in the early stages, such as discovery and preclinical. Early R&D requires them to be very nimble and being able to move from opportunity to opportunity. And large pharma ends up spending a lot of money and not having a very good success. And so that's why they prefer to buy or license innovation from smaller companies. On the other hand, we are focusing on the early stages. Our innovation and know-how takes care of the discovery part, and then we move on to the preclinical and phase 1 using the variety of standard protocols. And so as kind of visualized here on the slide, our interests with pharma companies start overlapping at the level of about phase 1, maybe phase 2 trials, as you can see here. So all we need right now is some extra dry powder to get us to that level, and also bring some other candidates in the pipeline up into the development. Oh, and also on the bottom, you will see the numbers, the percentages, and you can tell that we are right in and those percentages represent the success rate of moving from one stage to another. And so you can see that preclinical and phase 1 are right in the sweet spot as far as the probability of success of getting to the next stage. And so our expectations for the value of the program is shown here. So if you look at the curve, it represents the projected growth of the company's value. It could be company or the separate asset value. And the vertical axis on the left represents 100 million and 200 million as a company or asset value axis. And those points on the those little points here, I can show you with the mouse. So these points here, here, and there, basically represent milestones that drive the value of the compound upwards as it passes along the development path. The numbers that you see here, 2 million, 10, and 2025, basically reflect the specific case of our CGA17 development and the projected funding for each raise. And so to reiterate the earlier point, preclinical and phase 1 clinical represents the best opportunity to invest into the project due to a number of factors. First of all, it's a low cost relative to other stages. It's high probability of success in progress into the next stage, as we have seen earlier. It's basically use of standard protocols in the FDA mandated preclinical studies. And specifically for Lactocore, the use of peptides as drug candidates, as they represent pretty much the safest molecules around. So the largest increase actually in value typically happens after completion of phase 2, or the proof of concept in humans. As a rule of thumb, this is where usually biggest success stories of small biotechs happen. Ideally, this is exactly where we would like to be with LCGA17 and other candidates. So we project that we will have to come up with another $20 to $25 million to complete phase 2 in 2 indications for LCGA17. So where do we get the next round? Well, the source of this funding could be either equity investments, or it could be non-dilutive financing, such as, for example, licensing fees or the option deal fees with a larger pharmaceutical company. Remember, as I mentioned previously, at this stage, around phase 1 and phase 2 is where the interest of the pharma partners usually peaks. And we have a number of them interested in monitoring our progress. They're just waiting for us to get to that point, at least to the end of phase 1. And that's where we need your participation in this $10 million round. So finally, let me give you a quick overview of our team. Anton and Igor have started the company a few years ago, together with our investor, Mr. Babkin, who has a scientific background and has been with the company since inception and committed $3 million to this round. Igor leads the core team of young and talented bioinformatics experts, and Anton is our chief scientific officer with deep expertise in behavioral animal models and neuroscience. I have to mention that all the discovery and early development, a lot of what I talked about today, is actually managed by Anton and Igor, and this effort is mostly happening in Moscow, Russia. As soon as our candidate reaches the FDA mandated studies, we transfer all of the development activities to the United States. Doctors Pumal and Marshall are both industry veterans with experience in preclinical and clinical development respectively. Marshall has spent a lot of his time specifically in psychiatry drug development. So he's been an indispensable resource for us for planning out our clinical trials. As already mentioned by Lindsay, I'm a CEO of Lactocore. The company registered in Delaware and located in Ann Arbor, Michigan. I have a scientific background and a business experience, managing investments in venture capital firm, driving external partnerships in biotech companies, and managing early stage drug development projects in startups. We rely heavily in our work on our scientific and clinical advisors. Dr. Lasher is our leading expert in the mechanisms of antidepressant drugs. He's a key opinion leader in the GABA-A, depression, and related science. Dr. Pirine has been helping us with very tricky animal models of post-traumatic stress disorder and drug addiction studies. And Dr. Israel Librazon is a world-known expert in stress disorders and specifically PTSD, both at preclinical and clinical levels. He has been instrumental in guiding our research in the anxiety indication. And here on the final slide, I would just like you to leave you with a summary of this opportunity and my contact information. And finally, I'd like to thank you all for your time, and would be happy to answer any questions.
Thank you, Askar. And now would be the time that if you did have any questions, to please utilize the chat option so we can go ahead and answer those accordingly. Okay, Askar, it looks like we have one question. It says, can you comment on the status of your intellectual property?
Yes, sure. Excellent question. Actually, it's mentioned here in the investment highlights, but let me just give you a quick overview. It's a brand new IP. So we have filed our first provisional patent in March of 2019, so just over a year ago. And we've been filing the provisional since then, and now the first one was converted to the PCT. So we are working with a very advanced company for patent attorneys called Morgan Lewis. They're located in Boston in the United States, and we're using very professional and high-level attorneys for these purposes. So that's pretty much what it is about the IP.
Okay, perfect. And it looks like Gar Smith has a question. He says, sorry, I may have missed this, but can you conduct studies with a zebrafish? If so, what were the results?
Well, yes, absolutely. The zebrafish studies is part of our kind of discovery engine. Zebrafish is really good for very quickly screening out the best hits that are coming from the in silico development. So it's a funny thing that the zebrafish actually have a behavioral paradigms. So you can test them. You can look at different parameters, such as the time they spent on the bottom of the tank, or the top of the tank, light chamber, dark chamber, and how much time they spend in the school of fish as opposite to traveling or swimming alone. So zebrafish is a very quick and turns out to be very reliable way of quickly screening in silico hits. So for us, it's been a part of the early discovery engine. But obviously after that, we go on to rodents, and that's where we are right now for the LCJ17.
Great, thank you. And David has a question. He says, what are the side effects thus far noted?
The side effects that are noted so far, we have not really seen the behavioral side effects at this point. And for the toxicity effects, due to the fact that this is a peptide, we have done some acute toxicity studies where we loaded a lot of peptide in, way over FDA mandated dosing, and have not seen anything. So toxicity is very clean. That's not an issue. For the things such as sedation and addiction potential, as I reported already in the previous slides, we have not seen anything like that. We are, however, conducting further studies. These are not completed. There's more complicated studies for addiction that could be done and will probably be addressed by us. But so far, it looks really, really exciting.
Okay, perfect. Thank you. So Benjamin has a question. Are you also working with any third-party companies that assist and ensure you are meeting all FDA regulatory testing and phase requirements?
Can you repeat the question? Are we using any third-party
companies to assist to make sure that you're meeting all the FDA requirements?
Yes, we are. Well, at this point, we are absolutely relying on, first of all, our experience of our advisors, and a number of consultants. We have a regulatory consultants that help us with moving forward on the preclinical side. And at this point, preclinical is very straightforward. We have done it multiple times. We understand what it takes and what kind of documentation regulatory procedures have to be followed. One of the main things in all of that is to make sure that the CROs that we pick to conduct our experiments are FDA compliant, have gone through FDA audits, and can do the GLP related toxicity and all other studies as mandated by the FDA. So we pay extra attention to the CROs and to the compliance of those CROs with the FDA.
Okay, great. Let's see, Danny has a question. Have you tested BBB penetration of your drug candidate in vivo models?
Yes, absolutely. This was a little bit beyond of this presentation, but that's a very good question. I must add that we are developing peptides intended for intranasal delivery. So this delivery supposedly will increase the penetration of our peptides to the brain. We have done the experiments where we had the radioactively labeled peptide. So we had a tritium labeled peptide, which was intranasally injected into rats and mice. And we were able to track down its distribution, not only throughout the organism and to measure the PK, the pharmacokinetics, but we have also measured the distribution of this peptide in various brain regions of the rat and confirmed that, yes, we have definitely a very good penetration of the peptide into the brain.
What is the expected minimum investment amount, Askar?
It's going to be $500,000.
Okay, perfect. And can you elaborate on any potential exit opportunities?
As I mentioned on one of the slides here, success in phase 2 studies is what will definitely trigger a major partnership or licensing deal or a company or asset buyout. And so as a second opportunity, the company may list the IPO to raise funding and to provide liquidity, of course, if the market is favorable. And as the third option, the company using the model of generating the leads and drug candidates and driving them up to phase 2, we can keep doing this and generate attractive assets using our proven approach. And so we can just grow by using license fees from our partnerships, which will increase the company value, which will allow it to go IPO and provide the liquidity for the investors.
It doesn't seem like there's any other questions at this time. I wanted to thank everyone very much for joining the Family Office Network's webinar series today. Askar, thank you very much for explaining us Lactocore. I will be following up with everyone individually by tomorrow, but if you have any specific questions for myself or Askar, please feel free to reach out to us directly. We hope you have a great day and we look forward to speaking with you all soon.